Welcome. The following presentation was recorded at the annual Best Practices for Pollinators Summit, hosted by Pollinator Friendly Alliance and the Xerces Society. Please consider participating in the annual summit next year. You can find more about the summit at pollinatorfriendly.org/summit.
Great, thanks. Hi, everyone. So I'm Sam Talbot. I'm the Extension Educator here at Dakota County. It's a real pleasure to be here today. Thanks, everyone, for listening in. I'm really excited to talk about native plant propagation and share some of the lessons that we've learned here in Dakota County just over the past few years. So as the Extension Educator, I coordinate the native plant propagation program, and then support our master gardener volunteers. And what I found helpful when learning about how to grow some of these native plants at different scales is to visit similar plant production facilities and compare notes. So kind of today, I'll give you a bit of a tour and some of the insights and lessons that we have gathered just in the past few years here. So here on the left is our beginning greenhouse, growing plants in our southwest corner of our office building during the winter of 2018-2019. So we have a seedsters program where we provide volunteers with grow kits. So essentially our greenhouse is spread out throughout the county with volunteers in their basements and their windowsills. But then we started to invest in more staff time and resources to grow our own native plants, because we have a real need for them with our restoration that we're doing on county properties. So we not only increased the number of plugs that we produce each year, but also the number of the variety of species. And then we've also increased the number of volunteer hours and engagement. So why invest time and resources, staff, volunteer time to growing our own native plants? As opposed to just getting everything from the market. We still do purchase seeds and plugs from vendors, but we've been able to supplement quite a bit so far. So the more diverse a plant community is, the better that ecosystem can withstand, recover from climate extremes, from pests, disease, making them more stable and resilient. So if we can have a wider range of plant species and more diverse plant communities. And then growing locally sourced seeds for restoration efforts. And this, you might know as local ecotype, where the seeds or plants are well adapted to the conditions of the region from where they came. So we're able to collect quite a bit from remnant sites here in the county, or get seed collection permits from surrounding counties, and then sometimes actually get Dakota County origin seeds from vendors. So everything that we're growing in our greenhouse is from sort of hyper local provenance, and then everything that we're using in our restorations, we try to aim for within 150 miles if we're seeding something in or getting plugs. But as you may know, there's not much of that original prairie left. You can see here in the red on the map. So we want to be as efficient and as effective as possible when we're growing out these native seeds, because it's such a valuable resource. And then utilize species that are expensive or unavailable, things that we see as high quality. So, we're very interested in getting back into our restorations. Pidiomelum, esculentum, prairie turnip not only provides ecological value, but is also culturally significant. So we're getting that back into the bison range over at Spring Lake. And we were able to get a seed collection permit from Carlton Arboretum. Reintroducing that back into the county through propagation. And then special restoration projects. So we got, again, a seed collection permit from DNR to collect from SNAs. And then we're growing about 10,000 Viola pedata and Petitifida for for Regal Fritillary. So the host plant is prairie violets. Regal Fritillary are species of concern. And may possibly be at some point federally endangered, and with their ranges maybe shifting with climate change. So yeah, we're just starting that project itself, but we're able to grow, tens or thousands of, not tens of thousands of a particular species for a particular project. And then it's a growing program. So in 2019 is 700 plants. That was just our volunteers over the winter. Installed the hoop house with funding from the National Fish and Wildlife Foundation. That first year, kind of getting everything set, didn't have everything automated. We were still just building a volunteer base. One seasonal intern working on it, and then using pretty much the same amount of space, we were able to double it, so be more efficient. So we had just figuring out different ways to break dormancy really helped, and just getting better yield. So I'll talk about some of those techniques today. And then last year, so we rented even more space from the University of Minnesota, and then started wetland plant propagation. So we had an intern research and grow out a bunch of wetland plants this year, and we have pretty good success. So we attempted 119 species. Not everything grows well, but I think yeah, Fanny had talked about Cocoon. I know we're still trying that one too, but yeah, so we're certainly learning each year, and yeah, just trying to get a little bit better. And then volunteer engagement. So that's the other main goal of this program, is to get folks involved. And it really helps to increase our capacity, but also provides local residents with an opportunity to learn about native plants and how to grow them. I find, with the more popularity with backyard pollinator habitat, lawns to legumes, folks are kind of really looking for that hands-on experience to bring to their own yards and boulevards. And some of our volunteers have made it a goal to get involved with every aspect of the process. So from collecting seeds to planting them out, and then kind of continue that through the next season. So it is sort of a complex program. As we grow more plants, have more partners, more volunteers, but with any complex system, we want to find ways to be, again, more efficient, more effective. So getting higher germination rates, better data collection, more opportunities for volunteers to be involved. But that gets complicated pretty quick. So I like to just kind of look at each piece individually, kind of how it fits into the broader scheme of things. And then for the sake of time, I'll just focus on some more of those actionable components, things that you might be able to utilize if you are already growing native plants, or plan to grow them at whatever scale. And I'll just kind of quick tour of our growing facilities. So I'll talk about some of the infrastructure we have, and then the propagation, so the actual how we get stuff to grow. And then so we have two separate facilities. This is the original one that we hoop house that we built with the grant funding. This one is where we grow our upland species, and we have another space for wetland species. You could certainly grow them under the same space, but that just kind of worked out well with our setup. And then again, we're automating all of our irrigation, heat, ventilation, and then 2 zones in our shade house for hardening off plants. We want to harden them off for a couple weeks at the very least. A little bit more is better. And then we installed insect screening. So unfortunately high mortality of some of the native bees and dragonflies that would get in there. And the volunteers have been really great about catching them, and we take them down and ID them and release them. But that has really cut down on some of that mortality. And then our other facility for wetland propagation. So we start our seeds in this in those 10-20 trays for our wetland and upland species, and then for the wet loving wetland plants, we'll plant them or we'll get them going in these flood trays, and they'll stay submerged in water, and we'll replace that water and clean out some of the algae buildup. We're actually looking into putting in some more automated ebb and flow tables as well. And then, so since the remnant sites that we collect from are high value and can be sensitive to disturbance, we're establishing these propagation beds throughout the park. So we have some along trailheads. This is over at Lebanon Hills, and then built into retaining walls, we use some of this rain garden soil. It's a very dry, well-drained mix, so we can see some of the prairie smoke coming up there. So these have been really great. So we don't have to go back. So it's the same genetics that we're collecting from our remnant sites, but we don't have to go back to these sites and create more disturbances. And we can plant species from multiple sites as well. So hopefully mixing up that genetics. And then for our wetland propagation beds, we have the goal of these is to be able to easily remove them like you would sod and then install them. So maybe along a riparian or shoreline restoration. So that's kind of why we put this black plastic down here in that second step. And then you can see here on the right, our first crop that we started last year. So we're excited to see how well that does this year and start planting it up. And then, so I'll go through sort of the propagation process, highlight some of those, again, those lessons, some of the stuff that we've learned throughout each step. And that process really starts here with seed collection and cleaning. And so we don't really use any specialized equipment when we collect from remnant prairies or propagation beds. Simple hand collection, brown paper bags, scissors, clippers. This is and really fun with volunteers in the fall, do some late season plant ID. But we do create a unique collection ID number anytime we collect a species. So from a particular location. So that number will follow that seed lot from collection, cleaning, growing out in the greenhouse, out planting, and we can make sure that we're not over collecting from a site. So we can look back, see how much we collected, and then seeing how well, comparing different seed lots and germination rates. And then for restored prairies, so again, we're our protocol now wasn't always the case years ago, but we are striving to get our plant material from within 150 miles when we get it from vendors for our large restoration. So if we're getting a big seed mix or something. So when we know where that origin is from, so our newer restorations, we can go out with our, this is our, it's called the Reaper. So kind of walk behind combine. Up front, there's a little bit like a sickle bar, and then you can raise and lower the height. So we can get, we want to go in areas that are weed free, and then we'll kind of walk it through, try to get a mixture of forbs and grasses. And then that will go into our greenhouse in the fall. So nice and dry, dries out really good. And then I found last year just running it through a leaf mulcher, the seeds stayed intact, but you kind of break it up really well. And then we just take all of that stuff, the thatch, the seeds, and we'll spread that out after a burn or in areas that need some sort of, need a little bit of enhancement. And then cleaning. So I find cleaning sort of worth the hassle for a couple of reasons. So, you know, often weevils or insects will get into, especially some of these legumes, So you might think that all of these are viable seeds, but come to find out that they may have been predated on. So just knowing that, or getting down to the actual viable seeds is really good. And then also for that, again, that seed to soil contact, not only for propagating, but if we're spreading that out into some of our restoration areas. And then the couple tools that I use quite a bit for kind of smaller scale, some of the stuff that we're dealing with, hand debearder up top. This one's actually, I think they're made out of conveyor belt material, so it's rough enough, but not too rough to hurt the seeds. And those are a little bit more expensive. So I've just been making these abrasion boards, really cheap plywood 2x4 and anti-slip tread tape. Just get at the Menards, Home Depot. And those are really great because we can make a bunch for a volunteer event. And you can certainly use sandpaper if you're doing legumes or something like that. And then once you kind of get everything separated, I usually run it through a column of sieves. Here, what's nice is the seeds from a species will often be uniform size. So they'll end up in one of these as the sieves get smaller in diameter, they'll end up somewhere in that column, hopefully. But often they'll be mixed with still be mixed with some of that chaff. So this is one of the zigzag seed separators that I made. You can find the plans free online. It connects to a vacuum here, creates pressure, and then you drop your seeds in through the funnel. And then down here, all of the heavier, dense seed material will fall, and then all the lighter chaff will go to that side. So essentially, you're separating it by density. And this seems to work pretty well. Again, when your inert material and your seeds are the same size and you can't screen it out anymore. And then large scale operations. So we're not at this point yet. Not sure if we'll ever be at that at this scale, but this is a seed clipper. So larger facilities, larger operations will use these. And essentially it's kind of doing all of that stuff at once, where you're separating by size using screws, and then there's a blower there. So it's separating by density as well. And they do make some tabletop versions of these. And it all comes down to getting the right screens, and you can get screens that are specific to that species. All right, so you got your seeds, everything's clean. Then the next step is to get them to grow. And this is the kind of fun experimenting part that I get to do over the winter and spring. So typical diagram of seed germination for native plants. And I usually focus on two aspects when trying to get seeds to germinate, native seeds rather. So getting water into the seed, imbibition, or if it's a difficult to grow species and this isn't working, supplying the seed with directly with the plant hormone that's responsible for cell elongation and germination. And I'll go over just a couple examples of how we do that. So you're probably familiar if you've ever grown native seeds, germination codes. And for most forbs, they require some amount of cold moist stratification. And this is done with the using wet sand or vermiculite, and then for whatever is prescribed length of time. Then that's because native seeds are adapted only to grow under just the right conditions. So this mimics that first winter for them to go through. And for most species, this will work. So if you use the code or whatever suggested on Prairie Moon, it'll usually get you where you want to go. But with some of the plants, so again, we had about almost 120 species. Still a decent portion of them are going to fall into that category where either they'll germinate, or but they're not germinating reliably, or we just can't get them to break at all. So one of those was lead plant. Again, I mentioned for lead plant moth, I think we were growing about 5,000, and we're having kind of variable germination rates using the C10, the cold moist and scarification. But I built this steam box. We use it to sterilize our equipment. So it gets up to 150 degrees, and found that if you put the legume seeds, you sow them into moist soil, put them in the box, 150 degrees, steam treatment for 30 minutes, and we had really great germination rates. So that's our lead plant there. Before we were seeing a much spottier. And again, that can vary depending on the seed lot and seed viability. But this has been my go-to. You can also, there's hot water treatment that you can experiment with as well for the legumes. And then, yeah, so our, just a small portion of the lead plant that we grew, they take a couple of years to grow. So we're planting some more this year over at Spring Lake. And then prairie turnip. So I had mentioned that's one that we want to get growing, but we have small lot of seed, remnant seed. So what I'll usually do is purchase non-remnant, maybe from Prairie Moon, and then experiment a little bit with those. Finding that, again, getting water into the seed is the name of the game to start that process on that diagram that I'd shown. So with prairie turnip, yeah, steam treatment or individually clipping seeds isn't, I guess, as efficient, but it's definitely can be effective. So want to make sure you're not nicking the cotyledon. Yeah, so if we can't break dormancy using any of the like traditional methods or some of the, getting water into the seed, I've found that using plant hormones, naturally occurring plant hormones, work pretty well. So again, that GA3 is gibberellic acid. It promotes cell elongation and germination. Naturally occurs in the seed. Water starts that process, but you can purchase it in powder form. And I found that 125, 350, or 700 parts per million are good if you are kind of experimenting, trying to get some of these hard to grow species to grow. And then usually, and then soaking for 24 to 36 hours. And there's some there's information in the literature and guides online and exactly how to kind of work with this. But I find it, pretty straightforward for experimenting. And then you can also mix batches and freeze it in ice cube trays ahead of time, just to make sure that it's very clearly labeled, and not stored with other food or anything and other consumables. So one of the first ones that I had found, I was trying to get to grow reliably and a little bit faster, was gentian and chelone. So they have a 120 days requirement in the fridge. But if you soak them in growth hormone in the GA3, anywhere between 100 and 300 parts per million, we're able to break dormancy. And what we're finding is that one drawback is if you put too much, then they get very leggy and they're not going to be growing. They won't be growing well, and sort of fall over, and you're not getting as much of the root growth to kind of anchor them. But with these, we found, they grew really, really well. So again, it's kind of finding that right concentration. And sometimes that's experimenting, sometimes that's, found in the literature. There are native plant propagators who have kind of done a lot of this work too. So, so a lot of that information can be found out there of like where to start. And then, yeah, just a few more examples. So these ones all responded well. I'm actually growing in my office right now. So I lightly sanded with a 220 grit sandpaper for each of these, and then soaked them, because I found that breaking that seed coat just a little bit was enough to get the seeds to soak sink down into the mixture of GA3, and then soak them for 36 hours, and then just sow them as normal. So prairie blazing star, long bracted spiderwort, and then blue-eyed grass there. One that I don't have on here, but I also want to mention was the prairie violets. So that's how we found that we can grow. We grew about 2,000 last year, and we're hoping to grow about another 5,000. And that's the method that I'll be using. And then again, like plant hormones, they're not a silver bullet, just one of the tools. So with penstemon, we found that sure, it did help with germination. Over here is our GA3 treated, but then just giving it a little bit of scarification, and then using the C30, so the 30 days cold moist stratification, kind of with some of the hulls removed, help the water get in there while it was stratifying. So usually what I'll do is, if we're having low germination, is kind of run it through this suite of techniques and see how it works. One that I'm still working on, and if anyone has any suggestions, is rough drop seed. So when all else fails, just use natural stratification. If you're not familiar with winter sowing, highly recommend looking into it. It takes a lot of the guesswork out. And there's plenty of guides online, but essentially you're just kind of creating these small little greenhouses that overwinter outside in the snow, and then let nature do the stratification for you. So I'm trying some this winter right now. And then just a note about grasses. They're usually easy to grow, I guess, except for the pesky drop seed, but I looked into whether or not you should remove the hulls before sowing them. So when you remove the hulls, you can possibly damage the seed. And I did find with side oats grandma, it helped with increased germination, but otherwise, I mean, this is a little blue stem here. You can see unhulled and then hulled, pretty similar. So kind of saving ourselves that extra step of extra cleaning of the seeds. We can just sort of directly sow them. And these would probably even directly sow right into our larger containers. And then when to transplant seedlings. So since a lot of our transplanting is done by volunteers and conservation crews, we wanted to come up with a rule, like a guide for them of when we should be transplanting. So once we get them to grow, when are they large enough to go into our containers? And so our initial approach, transplant should begin when the first true leaves have expanded. In other words, when the shoots are ready, get these nice true leaves up here and they're ready to go. We were finding though, and here's one of our volunteers showing off a Sprengel sedge, that we should probably peek into the trays and see how the roots are doing, because sometimes you just can't tell from the seedlings. And then so here's just a few examples of that. So the graminoids, because they don't have true leaves, you kind of have to test, look in the soil and see where the roots are at. Same with these slow growing, so like downy gentian, especially when it gets hot in the greenhouse. Those aren't going to be, they won't survive if it gets too hot with those germination trays. So potting them up helps them at least put down some better roots and expand. And then the pale spike lobelia, so basal leaves, but right below, plenty of root structure. So we could have transplanted that a little bit earlier. And then that's kind of just a good rule, is that 4 to 7 inches or so of roots. And then we use these containers to transplant into. We use some short ones, some longer ones, so 5 inches, and then the other ones are about 7 to 8 inches. We really found there wasn't too much of a difference. So you can see, I think, same species here, long containers versus short containers when growing them. So like to actually develop the plants, what we're looking at now is monitoring after we plant them into restoration areas. So we can kind of compare and contrast. Does the extra roots help once they get planted out into the fields. You can kind of look down. Yeah, they kind of produce these pretty decent roots. They'll air prune at the bottom, and those channels kind of help direct some of the roots down. We did find though that if you're growing them on a natural surface, those roots don't care, especially the long ones, and they'll keep on growing. So using these, especially with some of those longer cones, getting them up off the ground onto tables really helped, and then they'll start air pruning. So a few benefits: get a little bit better soil moisture, less susceptibility to root circling, root rot, disease, and a little bit less transplant shock. Because
excellent talk, Glenn. We have quite a few questions for you.
Okay. Yeah, and I can finish up here. So just the outplant monitoring, I just want to mention that. So we had just 16 plugs per plot in these 4x4 grids, and then our plug survivability. So we were finding that we're getting about 30% mean mortality, which we sort of expected. So 70% survivability in that first year. And then hopefully, after that first season, we'll go back and we'll look at them and they'll be popped back up. So thanks.